Hello, my name is Jean-Jacques Kiladjian. I'm a hematologist in Saint-Louis Hospital in Paris, mainly involved in the management of patients with MPN. And today we'd like to discuss how can we measure disease modification in MPN clinical trials. And for that, we have one of the best experts in Europe and in the world, Professor Tiziano Barbui. Please introduce yourself.
Yes, thank you. My name is Tiziano Barbui. I'm professor of hematology in Bergamo and University of Milano, and my interest is MPN. And today I was asked to say my opinion about how to define disease modification in MPN. For instance, let's start with PV and ET. We started many years ago by producing some guidelines on criteria for defining the response in PV and ET. We thought at the time, it was 10 years ago, at the time that the best would be to have a complete hematological response, saying that normalization of blood counts in terms of erythrocytes, hematocrit, platelets, and leukocytes. But as soon as we published this, many studies documented that by adopting this criteria to assess the efficacy of a drug, say hydroxyurea, this was not correlated with critical endpoints, in this case with thrombosis reduction. So in other terms, you can reach hematological response, but you cannot reduce the rate of thrombosis in the follow-up. In the year after, we convened again, and we decided to add to the hematological response another part of the response that is in the absence of disease evolution. So at the end, we're defining 3 points that should be met to say we reached a response that can be a surrogate of disease modification in ET and PV. And at the end, we decided that in addition to hematological response and clinical response, we required also bone marrow reconstitution at integrum reconstitution, meaning that the bone marrow should become normal at the end of your test. And the third, the response, the molecular response. So the 3 should be met to define. In this case, I can think that the disease, we modify the natural history of the disease, but we don't have so far a demonstration that clinical trials have been produced with this particular endpoint, so that we don't know really what is this. But we are collecting many data now, particularly after the introduction of interferon. This data indicates that after some years of treatment with interferon, in PV particularly, the JAK2 is reduced very much, so that Professor Kiladjian demonstrated that if you reach less than 10% at the end of 3-4 years, in the absence of phlebotomy need, maybe this patient can discontinue interferon. So at this time, we are in the position to judge whether we are in the situation of disease-modifying disease. This is for PV and DT. Concerning myelofibrosis, things are very, very complicated. Also, in this case, ELN, in combination with the International Working Group, decided that a complete remission, including bone marrow remission, a partial remission, meaning everything normal, including symptoms, including blood counts, maybe that this can correspond to disease-modified definition. And if you are dealing only with a single item that you have corrected, such as anemia, for instance, you corrected anemia response, or spleen response, or symptoms response singularly, this does not qualify for disease modification symptoms, because these are only annotated as a clinical improvement. There are also additional criteria for that, so that surrogate, non-surrogate study, there has been a lot of papers dealing with what could be the real outcome to be reached in myelofibrosis to define disease modification. And a recent paper published in leukemia by Gianni Barrosi and Bob Gale indicated that probably the best way to identify the right definition and the right endpoints for a drug, just to have a disease modification, in this case, is the failure-free survival. Failure-free survival means that you can measure efficacy because you don't have a failure during this time. You can measure safety because the cause of safety can induce to discontinuation of a drug. And of course, the need of alternative is another failure in this context. So that this is what we are doing now concerning myelofibrosis. However, we have now other data, mature data after the comfort studies in myelofibrosis, after 5 and many years after the publication on these trials. And we are seeing now a prolongation of survival associated with some intermediate endpoint, such as splenomegaly symptoms and so on. So this is my introduction, say, for this discussion.
Very nice introduction, thank you. And obviously, as you mentioned, the best way to be sure that we are disease modifying is to improve the survival, in particular in myelofibrosis.
Exactly.
I would come back maybe to some pitfalls on the surrogates that you mentioned.
Yes.
First, the bone marrow biopsy. Of
Yes.
course, we know that this can be quite subjective. So I think in the frame of a clinical trial, we should at least be sure that the biopsies are centrally reviewed, blinded from the treatment received or from the patient clinical evolution on the treatment. And if possible, we saw at last EHA meeting, for example, that artificial intelligence may help to, let's say, more objectively measure some parameters like the degree of fibrosis, megakaryocyte morphology, etc. Do you think that this in the future should be applied, at least in clinical trial, to try to make it more standardized and more objective?
Yes, this is a great question, Jean-Jacques. I'm finishing the phase 2 clinical trial, and I have a bone marrow biopsies in polycythemia vera also. I organized a team of pathologists here in Italy, and centrally they review all the bone marrows in these patients. And they convene in a particular way, say that it's okay, and so on. And my suggestion was to be more objective in their definition by introducing the images and the artificial intelligence for deciding exactly the morphology of megakaryocytes and the deposits also of reticulin in the bone marrow. I asked these people, and recently I have to say that the question you are asking me now, I asked the same question to a group of pathologists who published recently the new guidelines for WHO. And they say time has not arrived now to apply this technique in clinical practice and in clinical trial. This is a very interesting, but we should have more experience now to combine. Also because, to me, at least, I am following this literature, but there are no studies comparing what is the group of results obtaining with the conventional histology versus the new artificial intelligence images. What is the difference? We don't know really whether the one sees things different from the other or vice versa. We don't know. So yeah, we can see. I don't know your opinion on that.
No, you're right. We have to make more clinical trials applying both the group of independent pathologists, humans, and the imaging in parallel, and see how they can compare, how they can improve one each other.
This is fundamental for that.
Another point, of course, is molecular response. You said that, you highlighted that. And I would say that, of course, we need to see a reduction in the driver mutation. Probably means that you can target one of the MPN clones. But we know now that the molecular signature or the molecular architecture can be very complex in this disease, in particular in myelofibrosis, with many additional mutations. Some of them are good, some of them are bad. And we have shown, us and others like Anne Muellerly, for example, that the series of mutations, for example, can affect the response to a given drug, like TET2 mutations or DNMT3A mutations that may reduce the response to interferon, for example. So what is now the best way to assess the molecular response in a, let's say, modern next generation clinical trial? Is it just following JAK2, or is it by NGS to follow a series of mutations? Should we go down to single cell sequencing to be sure of the architecture? What is your opinion on that?
My opinion is I am reviewing many papers, send them to review to be published in the literature. And many groups are dealing with this question you are asking me. I have to say that in this case, the complexity of the problems is such great that people cannot answer properly to your question. At the moment, we have to collect the data and then to see what happens. This is very important to know, because now the panorama is becoming wider and wider, so that you have driver mutation, you have non-driver mutation. You don't know which one of these are much more correlated with which outcome, which is the critical outcome associated with tattoo, for instance. We don't know. This is very important to know, because this can apply also to another situation that corresponds to the chip situation. As soon as we have a chip documented at the beginning or earlier of the phenotype of the disease, should I treat this patient to avoid the evolution towards the phenotype? There are so many questions that is very difficult to have a clear idea to answer your question.
Thank you. And maybe to conclude, one last dimension you didn't mention yet, that is coming more and more important, at least in the pathophysiology of myelofibrosis, is inflammation. What do you think about evaluating cytokines, etc.? Is it important in a clinical trial to measure the evolution of cytokines, for example?
Yes, I am very, very much in favor of that. I think the diseases we are dealing with, MPN, should be seen in this context of inflammation. This is documented by robust data. Many years ago, we published that a simple C-reactive protein, high sensitivity C-reactive protein, because people sometimes cannot repeat this data because they should use not the normal CRP that are usually tested during an infection or a fever and so on. But there is a particular CRP that is high sensitivity CRP that is associated with the vascular problems. And what we saw in the myelofibrosis, this is when it's high, associated with survival. So a critical endpoint and a very objective endpoint, so that the problem is, if I corrected this, can I reduce the mortality in these patients? Many things we learned from COVID disease. What is clear, if you reduce the inflammation during the acute phase of a COVID, you improve survival of these people. There are clinical trials that demonstrated that. So we can learn something from the COVID to answer your question, so that I am in favor of that. So we have to measure cytokines, and maybe studies should recognize some clusters of cytokines that could be related to the different groups of patients with the different outcomes.
Thank you very much. So just to summarize, I think we had many years with just clinical and hematological response, just normalize the blood count and improve the symptoms. But now we have many other dimensions, bone marrow biopsy, molecular architecture, and inflammation to measure in our clinical trials to show if we can correlate these changes to some clinical outcomes for our patients and improve our drugs. But still, this is a very evolving field, all of them, with many new data, new techniques also that allow to go deeper and deeper and more precise to evaluate this. So I think in the near future, we will have many biomarkers or surrogate markers of improvement that will allow us, hopefully, to improve the clinical management of our patients. So thank you very much, Tiziano.
Thank you. Thank you, Jean-Jacques.